Good day, this is Professor Will, CBMD PhD. And today's Thursday, November 25th, 2021. It is 7:22 and 42 seconds, p.m. Eastern Standard Time. Today's research, phages. So let's hop to it here. Phages kill dysentery-causing bacteria and reduce virulence in surviving bacteria. Date November 19th, 2021. Source: American Society for Microbiology. Phages are viruses that infect bacteria and can also be used to treat human infections. However, as with antibiotics, bacteria can readily evolve resistance to phage attack, highlighting a key limitation to the use of phages as therapeutics. Now, researchers have shown that the naturally occurring phage, 811 kills Shigella flexneri, a major cause of dysentery sub-Saharan Africa and Southern Asia, and selects for phage-resistant mutants with reduced virulence. That serendipitous finding results from the fact that the phages use of a particular surface receptor on the bacterium called OMPA as a portal to enter and kill S. flexneri. Means that bacteria that escapes a phage's attack will either lack OMPA receptors or that any remaining receptors will have mutated in ways that reduce virulence. Researchers sought to discover a phage that was naturally capable of binding to outer membrane proteins, S. flexneri, responsible for virulent cell-to-cell spread of this pathogen in the human intestine, hypothesizing that evolution of phage resistance should alter or eliminate this virulence factor protein, a biomedically useful evolutionary trade-off that improves upon standard phage therapy approaches. The researchers pursued phage therapy against S. flexneri because that bacterium was already resistant to conventional antibiotics. Additionally, this pathogen is active primarily in low-income countries, where antibiotics are expensive and often unavailable, and clean drinking water is scarce. Phages might even be useful for treating water sources by selecting for avirulent S. flexneri. The investigators began this project not knowing whether or not a phage existed that can kill S. flexneri. They assumed that such viruses had naturally evolved and could be isolated from environmental samples. And to increase the odds, researchers chose to search in a geographical region renowned for its extreme microbial biodiversity. Clearly a long shot, but apparently a reasonable idea, because this effort was successful. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.